Welcome to this tutorial we will be doing on the Golgi apparatus, an intracellular organelle that has a very specialized job and by no means is boring. So we have our eukaryotic cell here with its nucleus and its endoplasmic reticulum. So we'll write that down, nucleus and endoplasmic reticulum, and we need to know that because our Golgi is closely associated with the activity of our endoplasmic reticulum. So we have these two structures here, and we have our Golgi apparatus sitting close by, and it's got this strange interwoven tubule kind of shape that has vesicles coming off it in almost every single direction. So we're going to write down that it's an organelle composed of many membranous sacs or tubes, and it also has many vesicles. But like always, we can't really get a very good picture of it from above just like this, so we're going to zoom in. And actually quickly, just before I zoom in, I'll draw a couple of those vesicles surrounding the Golgi apparatus just so we know they're kind of hanging around there. So we've got a vesicle here, a vesicle over here as well. And now we're going to zoom in. So we'll draw our little magnifying glass and we're going to have a closer look. So what we have here in close proximity to our endoplasmic reticulum is our Golgi apparatus, and we can see that apart from having the vesicles coming off in all directions, it actually looks quite similar to the endoplasmic reticulum. And just like with our endoplasmic reticulum, we're going to call those infolds of the Golgi apparatus's membrane cisternae. So we'll draw that up here quickly. So these infolds in this membrane structure of the Golgi apparatus are called cisternae, exactly the same as our endoplasmic reticulum. But this is pretty much where the similarities stop between these two. And something we're going to find with the Golgi apparatus is it has two faces, the first of which being the cis face or the receiving face. And we're going to find the cis face of the Golgi apparatus facing toward our endoplasmic reticulum. And our second face will be the trans face, and we're going to find the trans or shipping face pointing towards the plasma membrane of our cell. Now we can actually talk about what's happening within our Golgi apparatus. So what it's going to be doing is modifying and concentrating all of the proteins and lipids that are going to be entering the Golgi apparatus from transport vesicles that come from our endoplasmic reticulum. Now let's draw this process so we get a better idea of what's going on. Let's say we've got this transport vesicle here that's budding off from our endoplasmic reticulum, and it's delivering newly folded proteins toward the cis or receiving end of our Golgi apparatus, and those proteins are then going to be modified and concentrated within that Golgi apparatus. Now something else that will happen is the newly folded proteins or lipids or anything like that are going to be tagged for delivery. So if we say this is a protein, the Golgi apparatus is going to put a tag on it that says, let's say it's going to the liver. Your Golgi apparatus puts that tag there and gives it an address, gives it an endpoint, tells your body where it's supposed to be going. And it's going to be sent off in new vesicles that will bud away from the trans or shipping side. And we have a few different types of vesicles that come off our Golgi apparatus, so we'll just point that first one out. We have the ones that are going to contain that new protein or fat or enzyme or anything like that, and they're going to be called secretory vesicles, and they're then going to go toward the plasma membrane of our cell and exocytose their content into the bloodstream or the surrounding tissues to be delivered wherever it needs to go. And the second type of vesicles that we have coming off our Golgi apparatus are going to become our lysosomes. So they're going to contain inactive digestive enzymes that have been created within our cell, and they're going to bud off here into this new vesicle, which we will call the lysosome. And we'll talk about the lysosome in a whole tutorial by itself. So we've got the lysosome here. And our third or last type of vesicle that we have budding off our Golgi apparatus is one that's simply going to contain phospholipids, and it's going to join our lipid bilayer of the cell. So it's going to become part of the cell membrane. And I'll just draw some plasma membrane here so we can get that picture really clearly in our mind. So our Golgi apparatus is going to have a vesicle that will incorporate itself into that plasma membrane to replace any of that phospholipid bilayer whenever the need arises within our cell. So we can see that we have lots of vesicles doing several different jobs and are delivering contents and marking it for delivery. And that's why we call the Golgi apparatus the post office of the cell, or the sorting center of the cell. So with that, let's review quickly what we know about the Golgi apparatus. We know it's an intracellular organelle composed of many membranous sacs and is going to be releasing vesicles with all different contents. It has a cis and a trans face, so the cis being the receiving, the trans being the shipping face, and it's going to modify and concentrate proteins and lipids that are coming from the endoplasmic reticulum and tag them for delivery around your body. I hope that's made things a little bit clearer for you. As always, thanks for watching, and I'll see you again soon.